Morning, everybody, and welcome to Grand Rounds. Usually the attendance of Grand Rounds relates to the topic, and I thought this would be a topic where the place would be overflowing because it's weight regulation, but I guess everybody's in such good shape that they don't have to worry about that anymore. Next week, we're going to have a case conference going to be presented by one of our child fellows, Jessica Eisenberg. When I first looked at this, I thought it said Jesse Eisenberg, and thought maybe we were going to have the social network here. And the discussants will be Anne-Marie Albano and Dr. Boris Rubenstein. And I just learned that Dr. Rubenstein, who's going to be discussing this week, actually mentored our Grand Round speaker when he was a resident, a pediatric resident in Boston. A couple of announcements. We had a good week this week. Meg Haney got a 2-year, $3 million R01 from NIDA for non-metabolized, well, it's a long title, but a treatment for cannabis use disorder. Alex Harris, who's a mentee of our former faculty, Josh Gordon, now at the NIMH, received a KO8 award for VTA nucleus accumbens circuit dynamics underlying stress susceptibility. I'm sure Josh had to recuse himself, and Alex got it on his own merits. Jonathan Javich got a 5-year, $3 million R01 from NIDA for probing mechanisms of amphetamine action on plasma membrane vesicular transporters. And Victor Luna received a 5-year, $650,000 KO1 from NIA for impact of increasing neurogenesis on cognitive deficits related to aging. In addition, our residents who last year won the Mind Games, which is like a psychiatric Jeopardy that the APA stages, are in the finals again this year, so it's like March Madness. And Joel Bernanke, Wei-Li Chang, and Anthony Zogby will be representing us, and anybody who goes to the APA should attend. It's really a lot of fun, and it's good to sort of cheer them on, so we're hoping for a repeat. One other announcement, which will be coming out in a blast email soon, is that since Maura Rinn announced she was going to be accepting the chair at Duke, we have been involved in determining how she should be succeeded and have completed that process. I'm happy to announce that Dr. Jeremy Veenstra Vanderweel will be assuming the role of Director of the Division of Child and Adolescent Psychiatry. And Jeremy has asked Dr. Laura Muffson to assist him as an Associate Director in that capacity. So congratulations, Jeremy. It's a real pleasure to have our Grand Round speaker who trekked across campus from the Berrie building here today, Dr. Rudolph Leibel. Rudy got his undergraduate degree at Colgate, his medical degree at Albert Einstein. He then went to Boston, where he was a postgraduate trainee at Mass General and Boston Children's Hospital, and subsequently went to the NIMH for a fellowship in pediatric endocrinology, and then came back to Boston, where he was at Harvard on the faculty at MGH for several years before moving to Rockefeller University, where he was rose to the level of assistant professor, associate professor, headed a laboratory, and then in 1997 was recruited to Columbia, and now is the Christopher J. Murphy Memorial Professor of Diabetes Research and also the co-director of the Berrie Center for Diabetes, along with Robin. What's Robin's last name? Golden? Golan, Robin Golan. Rudy has had an extraordinary research career in which he's focused his work on the molecular physiology and genetics of body weight regulation in diabetes. And in that context has cloned and characterized a number of genes in the pathways that regulate body weight and metabolic disease susceptibility. He's, in the course of his career, received many honors recognizing his prowess, as you might expect. He was elected to the Institute of Medicine, now the National Academy of Medicine. And interestingly, I see he's given 3 different Ray Kroc lectures. So Ray Kroc was the guy who made McDonald's what it is, And McDonald's, of course, was the kind of basis for that documentary Super Size Me, which is interesting. So it's interesting to see how Rudy sort of reconciles that with. But I won't say anything more about his scientific accomplishments, which you'll see when he gives his talk. But the other thing to say is that we've, and it's not just me, it's many people in psychiatry, have very productive and congenial or collegial interactions with Rudy collaboratively. The eating disorders group, Tim Walsh and his colleagues have worked with Rudy extensively. I and several people who've worked with me have collaborated with Rudy on looking at the metabolic and weight gaining effects of psychotropic drugs. And there's a lot of smart people at Columbia, as you well know, who are highly accomplished, and some of them are real mensches at the same time, and Rudy is certainly one of them. So it's a pleasure to have him here today to talk about molecular physiology of body weight regulation, physiologic and stem cell-based approaches. So please welcome Dr. Rudy Leibel.
Thanks a lot for the nice introduction. It's true, I've had very fruitful and ongoing relationship with people in this department around primarily studies relating to various aspects of obesity, body weight regulation, and as was mentioned, psychotropic agent effects on body weight. So I'm looking forward to continuing along those lines. Today, I was originally thinking I would focus primarily on some stem cell-based work that we're doing, but I sort of changed my mind and decided to describe for you, or at least to rehearse for you, some of the work that we've done on systemic body weight regulation, and then at the end, show you one of the very recent applications of stem cell biology to the actually attempts to discover mechanism lying behind a very complex genetic disorder of body weight, the Prader-Willi syndrome, which probably many of you in this room have seen for reasons other than, or at least in addition to, their profound problems with body weight. So I want to start by simply reminding you of something that many of you are probably quite familiar with. This is the figure which in the lab we've often referred to as the truck of life or the fundamentals of nutrition. You can take your choice, but what this is a sort of quantitation of the lifetime consumption of a human being with regard to amounts of various substrates, and measured in this case in tons, just to make the point. These are accurate as far as I know, indications of the amount of fuel that fluxes through the body of a human over a lifetime, including another half ton of vitamins and minerals, and the consumption from the atmosphere, at least up until the present time. I don't know what the current administration will do about this, but 10 million liters of oxygen to make use of those substrates. And they flux through a 70 kilogram, just to take a sort of average male over a period of a lifetime, and end up coming out of that individual, or the exhaust, so to speak, is carbon dioxide, heat, work, and some waste material. The individual is comprised chemically of this sort of makeup, which is measured not in tons, of course, but in kilograms. And the major sort of energy consuming part of the body is really this 10 kilograms of protein, which shows up in the form of the brain and fat and so forth. And the point here is that there's a huge flux of fuel through a relatively, at least in terms of this truck, small biological entity. And the miracle of this system, I think, and I'm going to show you, at least try to demonstrate to you some of the mechanisms behind how this works, is that the 70 kilogram man doesn't become, in most instances, a 700 kilogram man or a 7-kilogram man, that there is some homeostatic processes at work. And if you do the math on this just a little bit, average adult, male a little bit more than females, takes about 900,000 or 1 million kilocalories of fuel as food per year, and as little as a 2% positive error, meaning ingestion in excess of what's used here for the generation of work and released as waste and carbon dioxide and heat, is equivalent to a 2.5 kilo, 5-pound weight gain per year. Now, some people do gain weight over time, that's pretty clear, but most people don't gain 5 pounds a year, year after year after year. So this suggests that the system is in equilibrium to some extent, or homeostatically controlled. And if it's within a 2% or 3% error, this is well within the range at which things like sodium, urea, and glucose are controlled. But we don't generally tend to think of this system as highly regulated as it is. And what I'm going to argue is that it is highly regulated. The reasons for this regulation I will come to actually in the last slide. That is why the body would want to regulate the amount of stored energy in it or keep itself in weight homeostasis. But for now, what I'm going to do is show you some of the mechanisms behind that. So here's a pretty classic slide. I could have made this slide from probably 100 studies that are in the literature, but this one's as good as any. This is a study done in the early 90s showing recidivism to obesity in individuals. These are obese individuals who were put on 3 different regimens. One is very low-calorie diet and behavioral therapy, low-calorie diet and behavioral therapy, and very low-calorie diet alone. To your glory, I suppose, the behavioral therapy does add something transiently to the effect of the low-calorie diet, but everyone after 5 years ends up pretty much in the same place, which is interesting in 2 ways. They end up back where they started. They don't end up here, and they don't generally end up down here. They go right back to where they started. Gradually, the slope of the curve, the slope of the line is a little bit different depending on the type of therapy that's imposed. But this is pretty much the story about dietary therapy of body weight in human subjects. When drugs are used in place of the behavioral therapy, you may get a little bit bigger dip here, and then again, the inexorable recidivism to obesity. This can be read, and I think it does reflect the fact that body weight is regulated. It's not easy to lose weight, but it's even harder to keep it off. And again, I'm going to try to show you some of the biology behind this. So it was mentioned that I moved from Boston to Rockefeller when I got interested as a young faculty member in the question of why obesity occurs and why it is so hard to study. And I worked with Jules Hirsch, and he and I ultimately ended up, as was mentioned, running a lab jointly together before I came here. And here's one of the first attempts that we made to try to look at what might be happening in weight-reduced individuals. And these were data that were available through the Rockefeller University Hospital that had been collected in studies of individuals who had been put on various diet formulations. These were liquid formulas primarily, and caused to lose or gain weight, not so much in terms of interest in obesity, but it related to cholesterol homeostasis. This was work that the great pioneer Pete Ahrens had done that led to some of the most sort of important insights into cholesterol metabolism. But what we did was we took a look at these individuals when they were obese and after they had lost weight. These are the same individuals. And then a group of matched controls who were not obese and had not lost weight. And these were readily available through the data that Pete had collected. And what I'm showing you here is a first attempt to get an idea of how many kilocalories per square per 24 hours at the energy regard. And here you can see the obese range, something like 20 to 45, brought them into the Clinical Research Center at Rockefeller, fed them liquid formula diet like the one that Pete Ahrens had used until they were fully weight stable. This means within per day something like 10 or 20 grams. I mean, we're talking about really titrating them to body weight stability. Did a series of studies on them, some of which I'm going to describe to you. He gave them 800 calories a day so that they dropped to 90% of their starting. If we count the ones we did at Rockefeller and the ones here, we did something like 110. So this is like pterodactyl level food intake. I'm not saying this in a pejorative sense, it's physics. They are bigger, hence they need more energy, more fuel. They don't need more than lean people, they don't need less, they need the same amount proportional to metabolic mass. And here are the lean people, if you want to use them as these are non-obese. So here's the take-home message from this first sort of set of studies that I just showed you. You might have assumed before I showed you this that if you were to reduce an obese person down to a lean person, that you would be able to predict their energy expenditure by simply knowing this regression line, and whatever loss they had of lean and fat mass, you'd then be able to simply extrapolate to this plot. And that's not what happens. I already showed you this. What actually happens is this. So if you reduce the body weight of an obese person, their energy requirement now falls below this regression line when they're at usual weight. And for lean individuals, the response is exactly the same. It's not as if obese people are doing something physiologically that is unique to them. Their response is exactly like a lean individual or vice versa. And this is the conservative mechanism that I mentioned to you before, that will lead, at least if unopposed, to weight regain. And in the data that we have, and some of which I will show you, we refer to this difference here in a mathematical sense or statistical sense as a residual. It's the difference between what you would have predicted based on this line of identity at usual weight versus what you see in the individuals when they are weight reduced. So the question is, what's going on here? What is the mechanism for this? I showed you this from high altitude. So to explain this to you, or at least to show you some of it mechanistically, I need to, or at least I think it's appropriate to back up a little bit and give you a little background to what is known or was known about the sort of clockwork of the control of body weight in mammals. And this goes back to some experiments that were done by Hetherington and Ranson in the 40s, Brobeck in the late 40s and early 50s, that established the idea of a set point. And we're going to come back to that because obviously what I'm showing you is looking like there's a set point that is signaling the central nervous system that body weight has been reduced and leading to these compensatory changes. So in the 40s, these following experiments were done. Lesions were put into the ventral medial nucleus of the hypothalamus of a rat bilaterally, located just about here. And what you get when you do that to a rat is once the rat wakes up, it is tremendously hyperphagic. It will eat 3 or 4 times the number of calories it would otherwise want to eat and will gain weight over a period of time and double or triple its weight versus the control, and then level out or grow at the sort of slow rate that's normal for rodents. And if you take that animal and overfeed it by putting a tube down its nose and giving it extra calories, its body weight will go up. And if you then release it from the overfeeding, the body weight will go right back down to its new higher body weight, to its new set point, if you will. And if you fast the VMH animal, it'll lose weight like you would predict. If you stop the fast, the animal will go back up onto its new higher isobar of growth. I could show you the figures from this. I just didn't want to take the time to show it. But this led to the idea there's a set point for body weight, and what's been screwed up in these animals is the set point has simply been set up, but now it's able to regulate around the new set point. And animals in whom the lateral hypothalamus was lesioned, same way, had absolutely the reciprocal response. They became aphagic or hypophagic, and if carefully fed, would then start to level off and regain their body weight, but now well below that of the control animals. But like the VMH animal, if you under or overfed this animal, it would act like it was trying to defend its new lower body weights. Hence the idea that set point had been interrupted in these two models, and that the animals were trying to now defend these higher set points, if you will. And the physiology certainly fit with that. Now, fast forward to 1986, when at Rockefeller, I got interested, along with Jeff Friedman, in trying to get our hands on at least a single molecule that was dispositive with regard to body weight. Obviously, if you stick electrocautery into the VMH or LH of a rat, you're screwing up a lot of molecules. And it was unknown at the time exactly what molecules or tracks were being messed up. This is an animal that is homozygous recessive for a mutation on chromosome 6. It was known for 30 years. It had been identified 30 years ago, but the mechanism was not clear. This is a homozygous recessive form of obesity, and this is one of his brothers. And we set out to try to clone the gene that led this animal to be hyperphagic. And there were a series of studies done that showed that this animal actually had a phenotype metabolically that combined hyperphagia with lower energy expenditure. Looks a lot like a weight-reduced human being, but in this case, the animal is now, it's as if the animal is trying to regulate at a higher body weight. So we set out to clone this gene. Let's see, where are we? Here. And this is what came out of that. We cloned a gene called leptin, which it turned out was produced almost entirely by adipose tissue, and which signals the brain with regard to the amount of fat in the animal. And the reason that this animal gets so fat is that the brain of the animal is unaware of what you and I can see quite clearly, which is that it has an obesity problem. The brain is not detecting the signal from fat because there is no signal from fat, and hence the animal keeps eating as if trying to restore body fat because it's not using mirrors to gauge its diet. So here's, again, some of the clockwork that was discovered as a result, in part, of the discovery of this molecule. So it turns out that leptin is secreted into the blood, along with other hormones, such as insulin and ghrelin from the stomach, and these interact with the hypothalamus, or in the hypothalamus, as I just showed you earlier. This is part of the hypothalamus called the arcuate nucleus. It's right at the base of the third ventricle. There are two major cell bodies, two known major cell bodies. There are many other cells in this organ that we don't yet understand, but the AGRP NPY neurons secrete AGRP and NPY and increase food intake, and PALM-C is processed here to reduce food intake, interacting via these so-called second-order neurons that have melanocortin-4 receptors and NPY and other receptors on them. So again, just for purposes of showing you some of the machinery behind this, appreciate that there are actually reciprocally acting neurons in the hypothalamus that stimulate or suppress food intake, and leptin interacts with both of these. It suppresses the food intake and increases the activity of the POMC food intake suppressing neurons. And right at the end, I'll come back to this part of this pathway, showing you some of the work done with stem cells. But for now, this is really what you need to appreciate. There's a hormone produced by adipose tissue, leptin, that actually signals these brain centers with regard to levels of body fat. So once the hormone was identified, the gene was cloned, the hormone was identified, we looked because there was hope that maybe what's wrong with obese people is they just don't make enough leptin. They're like the obese mouse. They're not sensing the presence of their own body fat. And here is a plot of fat mass versus plasma leptin in humans, and you can see that that's definitely not the case. Obese people make more leptin, as you would predict, or as you might have predicted, and females actually make more per unit of fat mass than males because of the fact that subcutaneous fat, which women have more of proportionally than men, makes more leptin than visceral fat for reasons we still don't understand. We would very much like to know the reason for this, because it would help in terms of some therapeutic approaches to the treatment of the disease for reasons you'll see in a minute. Nonetheless, despite this pretty clear demonstration that there was no leptin deficiency in obese people, couldn't resist, not I, but others couldn't resist the idea of blasting humans with a lot of leptin to see if maybe that would make their body weight go down. So here is one of the two major trials of recombinant leptin in humans, looking at both obese and lean individuals. Because one idea was, well, maybe obese people are just resistant to leptin, so they'll take a lot more of it, but once you get the level up there, they'll respond, whereas lean people would be very sensitive to leptin. And there are a couple of things to look at here. This is weight change over a period of time in which the obese individuals were put on a mildly hypocaloric diet and then given these injections for a period of 6 or 8 weeks. And what you can see is out here where the dosing is sufficient to raise the blood level of leptin tenfold, and we're talking about really industrial blood levels here, the effect on body weight is really minimal. As a matter of fact, the mean here is just slightly down, as you can see from this green line. And notice that the lean individuals who were given these doses really didn't lose much or any weight. So it's not as if they were hypersensitive and obese people were non-sensitive. None of them really responded therapeutically, if you want to look at it that way, to this level of administration of this hormone. And we'll come back in a moment to what I think is the reason for this. Meanwhile, here's an experiment that we did to try to gauge what leptin might be doing in circumstances of a weight-reduced individual, where we know that leptin goes down because of the loss of body fat, and to try to gauge what might be happening in the central nervous system as a result of a decline in body weight. So here are rats that were either fed for F, fasted for S, starved, and then starved, same animals or same model of starvation, but given low doses of leptin by injection, sufficient to put the level back up to where it was before they had lost weight. These animals lost weight, obviously, when they were fasted. And what you can see here is that in the fasted animals, as I predicted or showed you from that model, NPY and AGRP, the orexigenic peptides, are up in the hypothalamus. These are direct measurements of expression of the genes in the hypothalamus. And the POMC, which is the anti-orexigen, causes hypophagia, has gone down, presumably, at least in part, in response to the decline in circulating leptin. And when we put the leptin back into these animals, we were able to normalize the activity of these central nervous system mechanisms controlling body weight, suggesting that a lot of the hyperphagia of a weight-reduced animal and its decline in energy expenditure might be the result of the decline in leptin below the level that it was at before the animal was manipulated in that way. So here's a model that derives from that set of observations that, and some others that I didn't show you, that the hormone is secreted from fat in proportion to the size of the adipocyte. So big fat cells make more than skinny or small fat cells. And they interact with the brain in ways that I've just sort of schematized for you, the arcuate and the PVN that I showed you before. But leptin interacts, as you're going to see in a minute, in many other parts of the brain. But for purposes of sort of understanding here, that the physiology of this hormone is not in preventing weight gain, which clearly couldn't be done by those massive doses that were given to humans and similar experiments in animals, but that the physiology is actually in the other direction, so that what's being monitored in the central nervous system is not how high you can get in excess of leptin, but whether the level of body fat has dropped below whatever is physiologically appropriate for that animal, and that's what the hormone is being used for. It's being used to detect downward deformity, if you will, of body weight in protection of the somatic fat stores that are critical to survival and reproductive function, as I'll come to right at the end of the talk.
There's no ceiling equivalent?
Say again?
There's no ceiling equivalent, so if there's like a threshold, doesn't like to drop below, there's something.
Could you repeat the question?
So the question is there a reciprocal of what I'm describing? What I'm describing to you is a system that prevents the reduction of body fat. Is there an equivalent system that prevents excess gain of body fat? And this is a topic actually of quite intense investigation in our lab. It's not leptin that does this, but if you hook an obese leptin deficient animal by parabiosis up to a lean animal, it acts as if there's something in the circulation of the obese animal that's trying to suppress the body weight of the lean animal. We're trying to figure out what that is. Whatever it is, and it may very well be there, it isn't strong enough to prevent an epidemic of obesity. So I think there is something there, and we're looking for it. I'll come back to that in just a second. It's a good question. So think of the system, just for sort of purposes of thinking about it or conceptualizing it, as a series electrical circuit in which leptin, in this case body fat, generates a current, which is the hormone in the circulation that is proportional to body fat, and it acts in the central nervous system through a series of resistors that are dependent on things like allelic variation at the leptin receptor, at the melanocortin-4 receptor. All of you, I'm sure, are aware that there are humans walking around with mutations, inactivating mutations of these two receptors that are very obese. Here I'm not talking about annihilating mutations, but just subtle variation in the level of the expression of these receptors and things like the cannabinoid receptor that determine how much current it takes to get through this series of resistors, if you will. And individuals get fatter in order to be able to close the loop. I mean, that's the analogy here. And there are clearly other components of the system, such as the other neurons that are in the brain, throughout the brain, that are talking to each other with regard to status of body weight, and glial elements that talk to those, and effects of diet and other cytokines, for example, that influence the receptivity of the system. But just for purposes of thinking about this, think of it as grossly as a system, a series circuit in which leptin is actually providing the driving current, and whatever is going on in the central nervous system that increases resistance to this system will cause an individual to get or to become more obese in order to close the loop, so to speak. So here is this idea shown in a little bit different way, but now consistent with some of the, or incorporating some of the other things I showed you. So this is the question of what happens when you lose weight in a system like this. So the purple horizontal lines here are equivalent to what one might consider a set point. So this individual who's got more body fat has a higher set point. We call it a threshold because for reasons that we're going to come to, we think that the major action of this is when the body weight or body fat or the signal drops below this threshold rather than exceeds it, relative to one of the questions that was just asked. Here's another individual who's leaner, who has a lower threshold or set point. The set point itself is determined by genes such as those that I mentioned to you before, by developmental influences. I'm going to show you one example of this in a moment, meaning pre and postnatal effects on structure in the brain, imprinting of genes and metabolic imprinting of the pathways, the circuits themselves, and obviously the environment. So what distinguishes these two people is that they have different thresholds or set point for body weight. And they will defend them in the following way. If this individual's body weight is therapeutically reduced down to this amount of body fat, the signal, as I showed you before, is proportional to the amount of body fat, so that now this individual at this level of adiposity cannot produce a signal which is sufficient to cross the threshold. And if the threshold is not met or crossed, the brain senses this as a deprivation state and sets off the compensatory responses in terms of energy expenditure and drive to eat. We'll come to the actual details of this in just a second. On the other hand, if you increase the level of this hormone by injecting it, as I showed you before, the body doesn't seem to be set up to respond to this kind of signal, at least not from leptin. There may be other signals that I mentioned or in response to the question that was just asked, but leptin itself doesn't seem to be important in terms of driving weight down. It's a signal that tells the brain whether body fat is sufficient for that individual relative to this threshold or set point. So based on this idea, here's another experiment that we did. I showed you already some data from this experiment where we lowered body weight in the way that I described to actually understand the physiology and behavioral phenotypes of these individuals. And then what we did in this another set of experiments is we administered this hormone leptin to them, not at supraphysiologic levels, but at doses only sufficient to put the blood level back to where it was before they started. So this would be equivalent, based on this figure here, to taking this individual who's now not making enough leptin to cross the threshold and just injecting into that individual enough to send it over the threshold by giving very low dose of the hormone. That's the experiment that we did. And here are the phenotypes that we measured. And I'm not going to go into deep detail about this, but want you to recall, with regard to the central nervous system, which is the home of the major regulatory elements of this system, that there are homeostatic parts of the brain. That's actually what I've been talking to you about primarily. This is hypothalamic and brainstem regions that play a critical role in reading out these adiposity-related signals from the mouth and from the vagus. But they're also obviously, and you guys are more expert than I am about these non-homeostatic factors such as emotions and mood, stress, and learning and previous experience that will influence actually an individual's response to food. So that what you see in an individual actually is in a sense, a vector that's resolving both the homeostatic and non-homeostatic factors that regulate food intake. So let me, just because this is a brain-centric audience, just show you one slide. We have a series of papers on this, but this will probably interest you to some extent. Here are the fMRI abstracts of fMRI signals from individuals at usual weight and weight reduced receiving either placebo, meaning a saline injection, or leptin. Individuals didn't know what they were getting because at these doses you can't really sense the fact that you're getting anything. And we looked at the responses of brain areas in response to food that was shown to the individuals while their heads were in the scanner, as opposed to things like shoes and rulers and stuff like that. Now, I think in some ways this could be described as advanced phrenology. I wouldn't put enormous faith in any of this, but I can tell you this, that there are regions of the brain in which the signals, the fMRI signals, are decreased, and others that are increased when an individual is given placebo at reduced body weight, and that these signals are reversed largely, not totally, by the exhibition of the hormone under the sort of experimental paradigm I'm showing here. And if you again parse these regions back to the map this figure that I showed you earlier, they are consistent with the idea that a lowering of body weight is activating these homeostatic mechanisms and interacting with the so-called non-homeostatic mechanisms in ways that would be predicted to drive energy intake upward, and that you can reverse a lot of these by the administration of this hormone. If you're interested in the details, they're in the literature. But here is a summary, a grand summary of a whole bunch of these experiments. So, and I've alluded to various parts of this, but not all of it. So if you look at the physiology of the weight-reduced state of a human being, this is a lean or an obese individual, their energy expenditure is about 300 or 400 calories less than you would predict from their new body size. The energy cost of muscle work is reduced. They are more efficient in terms of skeletal muscle activity, and I'm going to show you a little data about this in a moment. Their drive to eat is altered. They have delayed satiation. They have trouble perceiving. They underperceive the number of calories they've been fed. The neuroimaging changes I just described to you in very sort of brief terms, but you do get changes in these homeostatic and non-homeostatic regions. The autonomic nervous system is strongly perturbed under these circumstances. Sympathetic tone is reduced. Parasympathetic tone is increased. We've done this by actually infusing blocking agents into these individuals. The neuroendocrine axis is altered. Thyroid hormone is reduced. Leptin is reduced, obviously. Reverse T3, which is a starvation hormone, goes up. And in muscle, you see this increased chemomechanical efficiency, an increase in myosin heavy chain 1, which is the slow contractile myosin heavy chain of the sort of distance runner muscle, reduced glucose utilization, which is fast twitch, and increased use of fatty acids. So in this figure or list here, all of the phenotypes that are not in red are reversed by that administration of leptin. So virtually all of this appears to be at least strongly dependent on leptin, parasympathetic tone, and one of the sarcoplasmic calcium gates is not affected. And again, there's some interesting points to this that I'm not gonna belabor for you or at this point. So let me show you a little bit about the muscle, and since it's taken a little bit longer than I thought, I'll try to stop a little earlier. So if you sympathetically denervate muscle, and this is actually done in humans occasionally for reasons having to do with excess sweating, and you then exercise the forearm after sympathetic denervation, you get improved muscle energetics. This, again, is consistent with the idea that lowering of sympathetic tone can itself increase the mechanical efficiency of skeletal muscle. Hypothyroidism has the same effect. It increases the amount of the more efficient myosin heavy chain isoform and increases contractile efficiency. And you will recall that what I showed you here is that sympathetic tone is down and the thyroid axis is down, and that skeletal muscle work efficiency is increased. And that, remember, very early on, I showed you that non-resting energy expenditure is the major compartment for the effect of weight reduction on reduced energy expenditure. So now let me just show you a little bit of the, again, from high altitude, some of the muscle physiology of these individuals. So here are three different ways of looking at skeletal muscle efficiency or contractile efficiency, if you will. One is by bicycle ergometer. You put a person on a bike, you put an oxygen mask on them, and from the mask you can actually determine the rate of oxygen consumption and CO2 production. And what you do is you set the resistance of the bike so that they generate a fixed number of watts of power. And when you do this at low levels of power generation, you can show that the muscle efficiency of a weight-reduced individual is increased by about 20%. And the fraction of calories that come from glycogen or glucose as opposed to fatty acids is reduced by about 20%. So the muscle becomes more contractile efficient, and it does this in part by using a fuel which is itself chemomechanically more efficient. If you do this by magnetic resonance imaging, which is an entirely different technique, which is actually able to sense how much ATP is being used per contraction in muscle, you get almost the same answer looking at NMR spectroscopy, and the same answer with regard to fuel utilization, which NMR can pick up as well. This is really a remarkable concordance, I think. And then we've biopsied the muscle of these guys, and they have about a 20% increase in this myosin heavy chain 1, which is the more chemomechanically efficient, and an increase in that circuit 2, which is also leads to increased efficiency and is not altered by leptin. There's a reduction in phosphofructokinase, which is a glycolytic enzyme, and a preference for the use of oxidative enzymes such as COX. So if you look across this sort of three-way analysis of skeletal muscle, you get consistent results that point to the fact that a weight-reduced individual has gone, in terms of movement, from being like an SUV to a motorcycle. They just get more miles per gallon. And I'm going to show you a little bit more about this in just a second. So you probably, maybe, I don't know, if you're still awake, think this can't be true, or if it is true, it doesn't last very long. So the true part we'll come to in a moment. Here's non-resting energy expenditure looked at versus fat-free mass in individuals who are starting the study. It's up here. Here in the black squares are the same individuals when they've lost weight. I've already showed you these data in different form. They're more chemomechanically efficient. I didn't make it up. I mean, that's the data. But what happens if they stay down in weight? Now, I just showed you a figure right at the beginning that most people can't do this, but there are people who do, and they do it by very strict restriction of their food intake. If you've ever talked to a successfully weight-reduced person, they'll tell you this. They often still feel cold and uncomfortable, but they have to restrict their food intake. They often increase their physical activity. So would that relieve this metabolic, or if you will, skeletal muscle adjustment? And the answer is no. So here's a group of individuals who've had sustained weight loss for up to 5 years, and they're still on that regression line of an individual who's just at the end of one of those studies that I described. And what this tells us is that the body never gives up. And again, if you were designing an animal that would last and maximize its survival, you wouldn't want it to wait a month or two and then say, ah, to hell with it, I know I'm not fat enough, but I'll just give up. We wouldn't be having this one-way conversation at this point, I think, if that were the answer. We'd be in there with those big birds down at the Museum of Natural History. This, in one sense, is very disturbing, but in the other tells us that this is a piece of physiology that we should address, I think, in terms of therapeutics, and I'll say a word about that in just a moment. So let me come back to this muscle contractile efficiency thing and just show you two experiments that we've done more recently. One is to try to decide what fraction of that increased efficiency is actually due to thyroid hormone, which I've told you several times is reduced by virtue of leptin's effect in the central nervous system. You lower leptin, thyroid hormone goes down. So here is actually one of the studies I just showed you. So this is the leptin repletion study, individual at weight initial. This is efficiency, percent NREE expended as work. So higher is more efficient. This is the individual when their weight reduced on placebo, and this is weight reduced given leptin. And you can see that the black and the blue, we've restored the less efficient muscle phenotype. But here's the same experiment done on different individuals, in which we've done the same with thyroid hormone. And here you can see again the increase in efficiency, and here thyroid hormone, very low doses of T3 actually rectify the non-resting energy phenotype, suggesting that a substantial amount, at least, of the metabolic efficiency of muscle is being conveyed by the relative decline in thyroid hormone. This is tricky business. I wouldn't recommend that anyone go out there and start giving thyroid hormone to people who are weight reduced for all sorts of reasons that have to do with cardiovascular side effects and titrating the drug. But this is, in terms of the physiology, suggests that we're right about the idea that thyroid hormone is at least playing some role in this. We can get the same effect with leptin, which is reversing not only thyroid hormone, but autonomic status as well. So, I keep telling you, or I've said several times, that the skeletal muscle of a weight-reduced human looks more like a distance runner. It's got that myosin heavy chain isotype 1 and more contractile efficiency on a bicycle ergometer. So in an entirely separate set of experiments that have been done by people over the years, it's been shown that resistance exercise actually increases the myosin heavy chain 2, the fast twitch, glycolytic, less efficient muscle fiber type. So what we did is we actually resistance trained a group of individuals who were at maximum weight and who were at reduced body weight. Up here is just showing that this training regime did improve their ability, their peak torque, when we studied them in these various sort of exercise paradigms. It's just to confirm that we had the training effect that we wanted. But here what I'm showing you is the efficiency. These are the guys now on the bicycle ergometers before and after this resistance training. And again, I don't need to go into the details here, but what I want to show you is here they are at the beginning, either at maximum weight or reduced weight, and here they are after resistance training. You can see that what we've been able to do with these low levels of wattage is actually to reduce the efficiency of their skeletal muscle. That may sound in a sort of a teleologic sense like not such a good thing to do, but in fact, that's what you want to do to reverse the phenotype of more efficient contractile motion of skeletal muscle, and resistance exercise can do this. So again, if you were thinking therapeutically about the long-term management of a weight-reduced human being, you would want to, at least based on this kind of study, and you can see it at 2 different levels of wattage, you would want to encourage resistance training. So I'm going to just continue for another minute or two. I'm not going to show you everything I have on my small mind today, but I just want to make a couple of points, and then if you have questions, I'd be glad to answer them. Try to answer them. So what are the therapeutic implications of this? I've shown you now, and I think contrary to what people have believed, is that actually weight loss is a dysphysiologic or a non-physiologic state. It's not like, certainly with regard to blood pressure and diabetes and so forth, you can make a person better, but you're actually putting them in a hypometabolic and hungry state. And in that sense, they've been moved into a non-physiologic circumstance. And that if we could figure out a way, and I've shown you a couple of them, to put a weight-reduced individual back into a eumetabolic state, that is, restore the physiology without restoring the body weight, this would be a very important window into the ultimate treatment of human obesity, because the weight loss part can generally be achieved by most individuals with either diet or assistance with drugs or, you'll pardon the expression, behavior therapy. But they can't keep it down. So what we need is attention to how to sustain weight reduction in individuals who've gotten there by various means. And what I've showed you is, I think, some of the windows on ways that might be used to do that. Some of the thinking actually behind it. So again, let me end by just making the following point for you. I've shown you that this threshold or set point is whatever it is for a given individual. This is the leptin concentration versus fat mass. This is it here, goes up with, and individuals are located all along this continuum based on genetics and development and so forth. And in blue are lesions and events that could be interpreted, at least, as having lowered the threshold in an individual. Some aspects of anorexia nervosa, the LH lesion rat that I mentioned, some cachectic illnesses associated with cancer and other diseases act as if the individual was now defending in some ways a lower threshold. And there are other things that can raise the threshold, such as those VMH lesions, perhaps neuronal loss with aging, puberty and pregnancy act as if there were, at least in part, there were increases in the threshold, defense of higher levels of body fat. So also what I have shown you is that prolonged weight reduction does not lower the threshold. We cannot get the threshold down by weight reduction alone. But one very reasonable question is, could prolonged weight elevation raise the threshold? And I'm going to stop here simply because of the amount of time I've taken by maybe talking too slowly. We've done a number of experiments in animals that suggest that the answer to this is yes, that in fact, if you overfeed a mouse sufficient amounts of fat to raise its body weight and keep it up there for 6 months or 8 months, the animal will begin to defend the higher body weight versus a suitable control. So at least in animals, we've been able to show that you can do this. And we're now in the process of trying to understand what happens to the wiring and cellular makeup of the hypothalamus and other parts of the brain in response to an elevation, a prolonged elevation of body weight. One other, I'm not going to show you this, I am going to show you one other thing. It is known, and many of you in this room may know it, that leptin, in addition to some of the physiology I showed you, acts in the developing mouse brain. Almost certainly this is true in humans as well. Remember I showed you this arcuate nucleus here. There are critical periods of development in a mouse. The P here stands to postnatal day, 6, 12, 12, 8 to 10, in which the neuronal circuitry, the axons that go out to these other parts of the hypothalamus, for example, lateral hypothalamus, PVH, where the melanocortin-4 receptors are, the dorsal medial nucleus, the VMH. Leptin is required to form the circuitry. Without leptin, circuitry doesn't form, and the regulation of food intake is thereby deranged in some ways that I alluded to before. So let me just show you one other experiment that I think is very interesting in this regard, and it relates to the question of what is the developmental impact of excess body weight on subsequent, in developmental periods, on subsequent risk of obesity. This is just to remind me to tell you that we have actually been able to create a mouse in which leptin is driven not just by its normal physiology from adipose tissue, but in a way by a gene construct that when we feed the animal doxycycline, which is a tetracycline-like agent, it actually drives leptin production. So we can take this mouse, put doxycycline in the drinking water, leptin levels will go up, and as soon as we stop the doxycycline, the levels go down. So this gives us the opportunity to try to, in a sense, trick the brain of a developing animal into thinking that it's actually gotten obese, but without the confound of all the fatty acids and glucose and other metabolic derangements that go with it. This is just a pure play on what happens if your brain thinks you were obese at one time. And this is the last experiment I'm going to show you. So here's what we did. We took little mice, little meese, but just after they were born and up to 3 weeks, and gave them the mother's doxycycline. The stuff goes right through the breast milk. Remember, mothers are not supposed to take tetracycline when they're pregnant or breastfeeding because it stains the teeth. We're not that worried about the teeth of these animals, but the doxycycline here drives the leptin level up. And you can see here the exposure is very brief. So this is the equivalent to what would happen if you were chunky or got obese as a human, let's say, in the period where you were being breastfed. The animals are being breastfed for the first 3 weeks. And then we stop the exposure. There's no effect. The 2TG is the animal that's gotten, effectively, the others are controls, that's gotten this elevated blood leptin. So you can see after we stop, even when we're giving it, there's no effect, no major effect on body weight. But what happens if we now, when the animal is mature, it's 10 weeks old, we give it a high-fat diet. We let the animals see something that's very tasty and that they like to eat. And look what happens to the body weight of the animal that was exposed for just 3 weeks back here. It is hypersensitive to the high-fat diet, and you can see it even more clearly in the fat mass. I don't have to draw you a map about this. This is very intriguing because it indicates, or certainly suggests, that early exposure, and again, this from a demographic point of view, their epidemiologic point of view, this is not big news, that early exposure to adiposity, either in utero or early postnatally, may have profound long-term effects on risk of obesity. And here you can see evidence of it in these little mice. So what we're now trying to figure out, obviously, is what has happened to the wiring of the brain in response to this early exposure to elevated levels of this hormone without any of the confounds of the metabolic consequence. So let me stop here in case any of you have questions. I'd be happy to, hold on, let me just do one other thing. Just told you I would tell you this, so I'm gonna tell you, even if you don't wanna hear it. So you're all very polite. So I told you I would tell you why body fat is regulated. I've already shown you the data that certainly suggests that that's the case. But the reasons for this, evolutionarily, is that body fat is critical to reproductive integrity of the gonadal axis. Women or female or animals that have their body fat sufficiently reduced have the gonadal axis shut down. Anorexia nervosa is a outstanding example of this. But you'd want to be able to keep body fat above this minimum level so that reproduction would remain intact and so that you could survive under circumstances of lack of food due to what I describe here as environmental vicissitudes. And the question that Jeff asked about why you might want to keep it down below some maximum is primarily for predator evasion. And as I answered, it looks like there are systems in place that may serve that purpose in animals and humans. But if you consider what evolution has had to work with, in other words, what have been the major problems for human and mammalian evolution, this has been the bigger issue rather than trying to keep body weight down simply because access to sufficient food to get obese was almost certainly not widely available over sufficient periods of time. So you would predict, as I have shown you, that the defenses against the loss of fat mass are much stronger than those against gain. You see it in the physiology, and you also see it in the genetics. So when we look at the genetics of the system, about which I haven't spoken to you very much, again, what we see is primarily CNS genes whose primary role is to keep food intake up and energy expenditure down under circumstances of restricted access to food. Mike Rosenbaum did most of the human physiology, bioenergetics. This is Jules Hirsch. This is the young woman that did the mouse experiment that I just showed you. Dietitian here in the Clinical Research Center who's been enormously important in these studies. And for what I've shown you today, these are the two that did the resistance training in the experiments that I showed you. Thank you. If you have other questions or need to get a hold of me, that's where I am. If you see patients that you think might be of interest in regards to what I've talked to you about, get a hold of me. Thank you.
So, Rudy, that was a fascinating talk. I think the fact that evolution, as you showed at the end, the difficulty in losing weight and keeping it off is evolutionary makes sense. There's a little consolation for us, but I guess it helps to know.
That's what got us here.
That's right. That's what got us here. The physiologic set point that you're referring to, and you showed that some experiment at the end, a critical period of exposure, is that the main thing that defines it, or do we know what determines our set points?
So I think what determines our set points is a combination of things. One is the genes that we've studied extensively and about which I didn't say anything to you, but people who are inclined to maintain a higher body weight have subtle differences in the specific alleles of now upwards of 100 genes that have an influence on body weight. There can be developmental influences, and I just showed you one experiment that early exposure to higher body weight may leave an imprint in a metabolic sense. And in other studies that I didn't show you, we can do this to an older animal. So we can take an adult animal, force its body weight up, the equivalent of if it was living in New York City with great restaurants, hold its body weight up for several months, and it'll then defend that higher body weight. So this is a very complex system in which the sort of groundwork, I think, is laid by genetics. The twin studies and adoption studies suggest that maybe 40% of the risk variance for obesity is genetically conveyed, but there's another 60% that probably relates to many of these things that I either alluded to or showed you, and many of which we don't yet fully understand. This is not a simple problem, as you obviously are aware.
Yeah. Melissa and then Ralph.
This was really a phenomenal trajectory and explanation of work that's been happening over several decades. Thank you so much. One of the things you didn't touch on, which has been a really hot topic in the media, and I was just curious how this intersects with your work, is the role of the microbiome. And if you have any thoughts about how that relates.
So in the, we're sensitive to sort of fads in research. Maybe I shouldn't characterize it as a fad, but interest in the microbiome. And we've done two experiments. We did an experiment in mice in which we altered the diet and the body weight of the mouse to see what happened to the microbiome. And the experiment was done in such a way that we could control for weight perturbation versus diet. And the primary effect on microbiome was diet. So on a high-fat diet or a low-fat diet, and this I don't think is any great insight, the microbiome is quite different. But weight perturbation itself doesn't change the microbiome very much. And in the humans that I described who've had their weights perturbed on these liquid formula diets, we were able to do a version of that experiment where we didn't switch the diet around, but we switched their body weights around. And again, in a weight-perturbed human eating exactly the same diet, there's very little effect in the microbiome. So from those two experiments, which are published if you wish to see them, my conclusion is microbiome does not play a critical role in the compensation that I just described in terms of energy expenditure or food intake. It could be doing a lot of other things, but it's not doing that.
Ralph?
Yeah, two related questions. One is, I'm always interested in serendipity or clinical observations,
Yeah.
whether the choice of doxycycline,
Yeah.
how that came about. The only thing I know is some people on doxycycline have apparently delayed stomach emptying, and whether delayed stomach emptying affects sense of fullness. And the other question was, what drug is Lou Aroney using these days to help people diet?
Right. So the answer to the first question, the doxycycline was not selected by us with regard to any specific therapeutic impact on gastric emptying or anything. It is a construct which was created by molecular biologists a number of years ago, which can drive virtually any gene you want. We put leptin into the system, so to speak, but we could have put almost any gene behind this doxycycline sensitive mechanism. So the doxycycline has nothing to do with food intake effects of these animals because they're all getting doxycycline.
Doxycycline is part of the gene expression methodology that's
Right.
used.
It's just a mechanism to control gene expression. The fact that it's doxycycline is almost coincidental in this case. Now, Aroney is another story. Aroney is an aggressive, in a good sense, thinker about the regulatory processes that control body weight. And he could have gotten up in here and what he would say is, we don't understand enough about the neural circuitry that controls food intake in an individual. Clearly what Leibel showed you in terms of the POMC neurons and NPY, he would not gainsay any of that. But he would say that in any individual in this room, their response to agents that activate various parts of this mechanism are essentially still at this point unpredictable, with the exception of people who have clear mutations of one or another of these genes. So he's into empirical use of agents of, there are now 4 on the market that are licensed for this use that have actions in various of the pathways that are relevant to this, and one actually that's on the horizon, which specifically activates the melanocortin-4 receptor. And what he would say is he's agnostic in most instances with regard to what might or might not work, and he'll just keep trying different combinations until he gets something to work. And he would, I think, say, that's right. He's had circumstances in which there's been profound weight loss to 1, 2, or 3 agents in combination that he could not have anyway have predicted. But one thing I think he would tell you is if you stop the treatment, you're going to get the effect that I described to you, which is that they're going to go back.
You said that demonstrated how this physiologic set point is such a evolutionarily wired in and difficult thing to overcome. There's certain things that are able to overcome it, like we've seen, and we were working on the psychotropic drug administration. But apart from that, depression. So, weight loss, often profound weight loss with depression, presumably related to anorexia, occurs. Any idea what's going on there?
The short answer is no idea. But I think the fact that these are, that's why I tried to show you that sort of phony image, homeostatic, non-homeostatic. Clearly, the system has got hardwired characteristics to it, but it also has these non-homeostatic or hedonic mechanisms that influence food intake and affect and mood and all these things. And it's no accident that these things change in coordination with each other, but what it is that's actually ultimately doing it, particularly with regard to some of the psychoaffective aspects of this, I just don't know.
Jonathan?
Wasn't going to be my question, but that triggered a second question, which I'll ask first. Is the same thing true in terms of cancer cachexia? Is there any insight into what's changing molecularly that could be tapped into therapeutically?
The answer to that is yes, in the following sense. Again, I don't want to get too much into the weeds here, but there are pathways from the NPY AGRP locus in the hypothalamus to the parabrachial nucleus that is a glutamatergic signaling pathway. And if you knock out the NPY, AGRP neurons physically, not knock out the transcripts, but knock out the cells themselves, you end up with a mouse which is completely aphagic. I mean, the animal simply won't eat and will simply die of starvation, even given perfectly adequate amounts of food. There are now people studying this pathway very carefully for the purposes of trying to gauge whether or not it can be manipulated or whether it's altered in cancer cachexia in animals that have been transplanted or implanted with tumors, and whether glutamatergic manipulations of the PBH could actually be used to relieve this. So I think the answer to your question is yes, and it may turn out that some of those better understanding of the mechanisms for actually jacking up food, and to make people want to defend a higher body fat, could ultimately relate or be useful in terms of understanding of obesity. That degree of aphasia in those animals is absolutely incredible. I mean, it's that and an LH lesion, which was done almost 70 years ago.
You don't
just
think that
produce animals that won't eat.
You're not saying that those cells disappear in cancer. So it's some
No,
kind
I
of
think it's
therapeutic
probably
function.
some interaction of whatever the humoral release from some of these tumors is. And again, as you know, they're not monolithic in this regard. Some tumors have much more, and that's true in humans as well. It's just not understood. But my guess is that study of that PBH mechanism is going to be very helpful in this regard.
So my other question is, you said it, but you didn't sort of finish it. So clearly giving huge amounts of leptin in those studies failed, but everything that you measured except circuit 2 and one of the parasympathetic went away. So does this imply, it may not be practical, but does this imply that if you actually first fasted people and then gave them leptin, that they could in fact maintain their weight long term and be happy, and that you've solved their problem?
I think the answer is probably yes, at least in some of them. And that there actually has been some effort to test this in a more clinical setting, but the problem has been that higher doses of leptin have been used than simply replacement. And the system, as you can imagine, is set up so that if you actually drive the levels very, very high, the system shuts down. So it actually defends the body against just promiscuous loss of body fat. So high levels of leptin in a weight-reduced individual is not the way to do it. You need to give very, very low doses. And for whatever reason, there's not been, that hasn't been tested. Go figure.
But you tested them in
We tested it in a very highly
but
controlled
not in a
circumstance.
real life setting.
Correct.
Last question, I'm going to put you on the spot. So our dean has a book that came out, Too Much of a Good Thing. You're familiar with it?
I am, yes.
And do you think that his argument about evolutionary effects in the modern age are accurate?
I do. And in fact, when I talk to the medical students, for whatever reason, they still let me do that. When I talk to them in the quantitative genetics course, I have a slide, I could probably find it if I had enough time, in which I show a series of phenotypes that are currently medical problems that have probably at least part of their genetic background in evolutionary selection. Things like hypertension, the immune system being high. So yes, I agree with that. I think a lot of our current illnesses, not all obviously, can be accounted for, at least our susceptibility or differential susceptibility can be accounted for by evolutionary selections of one kind or another, just like this.
I'll tell him you said that.
Yeah, please.
Thank you very much, Rudy.
Thank you.